Hello friends, welcome back to Medico Srinjoy. Today we'll study a very important topic, that is the development and growth of teeth. First of all, let's start with the dental lamina. First, we need to understand the development of dental lamina. When the embryo is just 6 weeks old, certain areas of the basal cells, I repeat, the basal cells of the ectoderm rapidly proliferate than the adjacent cells, which leads to the formation of the dental lamina. Further, this dental lamina gives rise to the primary teeth, and the distal extensions of the dental lamina gives rise to the permanent molars. Further on, the successional lamina, which is later formed, gives rise to the permanent incisors, canines, and the premolars. Next, we need to understand the fate of the dental lamina. About 5 years old, that means from the 6 weeks old to the 5 years old, this dental lamina persists. As the teeth continue to develop further after the 5 years old, this dental lamina and the tooth lose their contact. But sometimes remnants of the dental lamina may persist and are called as the epithelial pearls or the cell rest of series, which are mainly found in the gingiva region. Friends, let's move on to the next part, that is the morphological stages of the development of tooth. This is the most important topic in our today's video. These morphological stages mainly of three stages. These are: first the birth stage, next we have the cap stage, and third we have the bell stage. Further on, this bell stage is again can be divided into the early bell stage and the late bell stage. The picture of the tooth germ is given. You may pause the video and see the different stages of the development of tooth. Next, we will read one by one these stages. Let's start with the birth stage. So what happens in this birth stage? In this birth stage, the enamel organ, which is shown in the previous slide in the picture, this enamel organ consists of peripherally located low columnar cells, and there is centrally located polygonal cells. I further repeat that in the peripheral location there are low columnar cells, and in the central location there is polygonal cells. Further, as a result of the increased mitotic activity, tooth germs gets formed. Next, let's move on to the cap stage. Let's study what happens in the cap stage. Unequal growth in the tooth bud, that is in the unequal growth in the tooth bud, leads to a shallow invagination on the deep surface of the bud, which leads to the formation of a cap stage. In this cap stage, there are mainly three types of cells seen under the light microscopic examination. These three cells are very important for the viva and the exam purpose. So friends, let's study this topic in detail. First, we need to understand that at the convexity of the cap and the concavity of the cap and the center of cap, there are three positions and there are three different types of cells. First, at the convexity of the cap, there is cuboidal outer enamel epithelium. I further repeat, in the convexity of the cap, there is cuboidal outer enamel epithelium, and in the concavity of the cap, there is tall columnar inner enamel epithelium. So in the convexity there is cuboidal outer enamel epithelium, at the concavity of the cap there is tall columnar inner enamel epithelium, and in the center of the cap there is a presence of very special cell which is polygonal stellate reticulum. So in the center these are polygonal stellate reticulum. These polygonal stellate reticulums are the star shaped cells. So let's move on to the next slide, that is what are these stellate reticulum cells. This is very frequent question in the viva, so let's study this topic in detail. These stellate reticulum cells have mainly two functions. First, it gives rise to a cushion like consistency. Okay, it's provide a cushion like consistency and support and protect the delicate enamel forming cells, that is the inner enamel organ. So first function is that it provides a cushion like consistency to the enamel forming cells. Next, we have the next function, that is These stellate reticulum cells, these stellate reticulum cells get collapsed before the formation of enamel, and so these cells helps in bringing the capillaries in the dental sac close to the inner enamel epithelium, and which provides the nutrition. So the next function is that it gets collapsed, which helps in providing the nutrition to the enamel forming cells. Next, let's study some transition structures in the next slide. These transition structures are very important in Viva questions. There are mainly 5 structures. These are very confusing, so let's study this structure in detail. So the cells in the center of the enamel organ. So I repeat, these cells are present in the center of the enamel organ they are densely packed, and these are transitional in forms. First, we have enamel knot. So what are enamel knots? Enamel knots are the knob-like extension of the cells. These are like knobs, so the name is given as the enamel knot. So enamel knot is nothing but a knob-like extensions of the cells. Next, we have the enamel cord. So what are these enamel cords? These enamel cords are the vertical extensions of the enamel knot. So to define the enamel cord, we first need to understand the enamel knot. Next, we move on to the enamel septum. So the question is, what are these enamel septum? These enamel septums are nothing but the enamel cords extends and the meet outward enamel epithelium, and this extension is called the enamel septum. I further repeat, enamel septum is nothing but the enamel cord extends and meet outward enamel epithelium. Next, we have a very important structure. What is enamel navel? Enamel navel is nothing but a depression present at the junction of the outer enamel epithelium and the enamel septum. So enamel navel is nothing but a depression which is present at the junction of the outer enamel epithelium and the enamel septum. And last, we have enamel niche. What are these enamel niche? These are the ectomesenchyme forming double attachments of dental lamina. So enamel niche are the ectomesenchymal forming double attachment of the dental lamina which overlap at the oral epithelium. So this enamel niche is clear to all. Next, we move on to the third stage, that is the bell stage. This bell stage is further divided into early bell stage and the late bell stage. Let us first study the early bell stage. So what happens in the early bell stage? Let's study. As the invagination of the epithelium deepens and its margins continues to grow, enamel organ assumes bell shape. So the birth stage gets converted to cap stage, and cap stage gets converted to the bell stage. There are mainly 4 types of cells seen under the light microscopic examination in this early bell stage. These 4 cells are very important, so let's study these cells. First, we have the cuboidal outer enamel epithelium. Okay, these 4 cells are very important, friends, so let's study this. First, we have the cuboidal outer enamel epithelium. Next, we have the polygonal reticulum. Next, we have the polygonal stellate reticulum. Third, we have the squamous stratum intermedium. This cell is new to you guys, so let's study this in next slide. And fourth, we have the tall columnar inner enamel epithelium. So except the squamous intermedium, the 3 cells are already present in the cap stage. So let's study what are these squamous stratum intermedium. This squamous stratum intermedium mainly functions as the protein synthesis unit and nutrient transport. So this stratum intermedium mainly regulate the formation of the enamel. So guys, let's move on to the next slide. Now the question is, as the stellate reticulum disappears, so what happens as the stellate reticulum disappears before the enamel formation? The outer enamel epithelium is laid in folds. So the folds helps in bringing the capillary loops, and these capillary loops provide the nutrition for the intense metabolic activity of the avascular enamel organ. Next, we have to study that as the inner enamel epithelium begins to form the enamel. So the inner enamel epithelium starts forming enamel. Then the peripheral mesenchymal cells of the dental papilla differentiate into odontoblast and it later form the dentin. So let's revise this topic that as the stellate disappears before the enamel formation, the outer enamel epithelium is laid in folds. Between these folds, the capillary loops provide the rich nutritional supply for the intense metabolic activity of the avascular enamel organ. Further on, as the inner enamel epithelium begins to form the enamel, the peripheral mesenchymal cells of the dental papilla differentiate in odontoblast and these odontoblasts later form the dentin. Also, we have to study that the dental sac show a circular arrangement of fibers and this resembles as the capsule. So let's study what are these fibers. When the root development occurs, these fibers get differentiated into periodontal ligament fibers. We'll study this in the upcoming lectures, that is in the periodontal ligament lecture. So let's see that the dental sac show the circular arrangement of fibers and it resembles as the capsule. Next, let's move on to the next slide and let's study the next topic, that is the advanced bell stage. Now guys, let's study what happens in the advanced bell stage. This advanced bell stage is characterized by the beginning of the mineralization and the root formation. So there is a characterization of starting of the mineralization and the root formation. Also, as the first layer of dentin is laid down, the inner enamel epithelium cells become functional ameloblast. Also, there is a reversement in polarity of this ameloblast, and in future this ameloblast gets formed enamel. So this outlined the future dentin enamel junction. So let's start the next part, that is the cervical portion of the enamel. In the cervical portion of enamel gives rise to a very important structure, that is Hartwig's epithelial root sheath. Let's study this Hartwig epithelial root sheath in the next part. Let's revise this topic that in the advanced bell stage is characterized by the beginning of the mineralization and the root formation. Also, this ameloblast is laid down and in future this ameloblast gets formed to enamel. So this advanced bell stage outlines the future dentino-enamel junction. Also, the cervical portion of the enamel gives rise to a very important structures, that is the Hartwig epithelial root sheath, also called as epithelial root sheath of Hartwig. So let's study about this Hartwig epithelial root sheath in the next slide. What is this Hartwig epithelial root sheath? This Hartwig epithelial root sheath plays a very important role. It plays a very important role in the determination of the shape, length, size and the number of roots. I repeat that the epithelial root sheath of Hartwig or the Hartwig epithelial root sheath plays a very important role in determining the shape, length, size and the number of roots. This Hartwig epithelial root sheath contains mainly two types of cells, that is the outer enamel epithelium and the inner enamel epithelium. So this Hartwig epithelial root sheath only contains two cells, the outer enamel epithelium and the inner enamel epithelium. Now let's study this topic once again for revision purpose, that is what are these Hartwig epithelial root sheath. These Hartwig epithelial root sheaths are very important structures and these mainly determine the shape, length, size and the number of roots. Also, these cells contains only two types of cells, the outer and the inner enamel epithelium. Friends, if you like my video, then you can download the PDF notes from the description box, which contains the total handwritten notes of this lecture in a PDF format in the description box. Next, we'll learn about what are these enamel pearls. It's a very important question often asked in the Viva and also in the examination, that is what are enamel pearls. These enamel pearls are nothing but these enamel pearls are formed from the Hartwig epithelial root sheath. So let's study this. If the Hartwig epithelial root sheath remains adherent to the dentin surface, they may differentiate into ameloblast and produce enamel on the root. So guys, this Hartwig epithelial root sheath remain adherent to the dentin surface, so it may get differentiated into ameloblast and it produces a enamel on the root. Such droplets of enamel on the root is called as the enamel pearls. So enamel pearls are nothing but the enamel on the root which gets formed due to the adherent of the Hartwig epithelial root sheath. These enamel pearls are mainly found in the areas of furcation of the root of the permanent molars. I further repeat that these enamel pearls are mainly found in the furcation of the root of the permanent molars. This ends our today's video, that is the development and growth of the tooth. If you like my video, then subscribe the channel to get more videos like this. Next day we will study the histophysiology in the development of tooth. It's a very small topic but often asked in a 5 marks or the 10 marks question in the examination. So we'll study this in the next part, that is the histophysiology in the development of tooth. If you guys like my video, then like this video and comment your queries. Share these videos with your friends and the share knowledge. So guys, if you are getting help from these videos, then subscribe the channel and hit the like button. So guys, thank you, keep supporting.